Hey guys, I'm Dr. Rishi Desai from Osmosis. I'm the Chief Medical Officer. I'm here today with Jesse VanWestrienen, who is the co-founder of Biomeme. He's a virologist, a molecular biologist as well, and actually worked on CoV-1, the virus that actually preceded CoV-2. So we're here with him today. Thank you so much for being with us here today, Jesse.
Hey, thank you for giving us the opportunity to visit with you today.
Jesse, can you get into Biomeme, the role of the company, what you guys do, and also how this test works and where it might work?
Sure. So at Biomeme, we develop a series of tools that allows anyone to isolate nucleic acids or DNA. We design tests to detect DNA from those samples. We make hardware that allows anyone to amplify that DNA, and our hardware systems communicate off of a smartphone app. So what Biomeme is about is enabling anybody anywhere to detect DNA, in short. And really what it's about is providing information from DNA so that stakeholders can use that information. In the case of a diagnostic for human health, that can guide treatment options, right? So Biomeme, we've been around for about 7 years. And we had a variety of different use cases we've been working on before this COVID-19 outbreak. A lot of the customers that we had are anyone from the US government who is looking at environmental surveillance. We have a wide variety of researchers that do field work where they want to detect DNA in the field. We've been working with some industry partners around manufacturing and industrial quality control. So it really runs the gamut of use cases and industries, and there's many more applications that are like that. Since the COVID-19 outbreak, however, we've been inundated with requests for people looking for testing solutions, and that includes the U.S. government and hospital systems and others. And so, really, it's going to be our main focus for the next, I'd say, 3 to 6 months, certainly, as we work to scale our manufacturing to provide those testing solutions. I'm going to give a brief demonstration of our products for your users, and I hope that it'll make a little more sense for everyone here. So our platform includes a consumable sample prep product. So this is a one-off use sample prep cartridge. It contains all the chemistry and buffer necessary to isolate DNA and RNA from a variety of sample types. We developed this cartridge here with a foil seal on top and also this sample prep column. It interfaces with a 1 ml syringe. And essentially the way it works is a user simply would perforate the foil and they would add a sample to it. In the case of SARS-CoV-2, that sample could be something like a nasal swab, nasopharyngeal swab, or an oral swab. Once that swab sample is placed into the transport buffer, it's added into that sample well right there. That sample well contains a lysis buffer. So what it's doing is it's breaking apart all the cellular material and freeing the DNA or RNA. The sample material that's in this column is a membrane that will actually bind the DNA from the sample, but leave all the other material in the liquid there. And so really the way you bind after adding your sample is simply pumping up and down. So we've designed our sample prep so that, again, really anybody can do it. It doesn't require any other lab equipment or specialized training. So I've been binding my nucleic acids to the column here. And next step is to simply move over into a wash buffer. So many of these sample types have a lot of different gunk or junk in there, and you need to wash them to remove any of that gunk on the filter. It's followed by another wash. This is actually a salt wash. So it's washing the membrane of the salts that were used to purify your sample. We have a drying wash, and that's followed by an air dry step. So now we're removing any residual buffers from that membrane. And the last step here is an elution. So the DNA that was stuck on the membrane is now going to come off the membrane. And your purified DNA or RNA is now in solution there. So that's it. As you can see, it just takes a couple minutes. If you're not talking to explain it, it's even faster. And now this last well there has your pure DNA or RNA. So really the next step in the process are the freeze-dried PCR assays themselves. What that is a real-time PCR assay that is contains all the specific targets that will identify whatever test you're interested in. So in the case of SARS-CoV-2, these assays actually detect two different gene targets of the SARS-CoV-2 genome and an extraction control target. So the way you work this is you simply remove that foil. You then are going to add some of your sample into the well. And I'll just do all three here. You cap it, and then it goes in the hardware itself. This is our Franklin real-time PCR thermal cycler. It's a 9-well PCR thermal cycler. What that means is you actually are using light to detect your various DNA or RNA targets. The hardware itself communicates via a smartphone app. So we have our Biomeme Go app here. This can communicate to the hardware device via Bluetooth. And the way that works is you simply start run. You're going to scan a QR code on that test pouch I showed you earlier. You're going to confirm your test. You can scan a ID, a sample ID. And then you will save your result, hit confirm, and it's going to walk you through how to power on the device and get the test running. So as you can see, that also takes only just a couple of minutes, and then you have your test running.
That's incredible. How did you guys get this together in just a few weeks? I'm so impressed.
Yeah. The initial design work is pretty quick these days. They released sequence from China very early on. It's publicly available. And there's tools these days that make it quite quick, very powerful computer tools to do your in silico analyses, et cetera. So the initial design really took a few days. And then nowadays, getting DNA synthesized is also very cheap. So the particular markers that are in the test can be synthesized within a week. You can also synthesize short strands of DNA from that sequence. You can pull any sequence from a computer and have it synthesized. That allows you to use to do development of these tests without any actual genomic or viral material, which was much harder to get hands on, at least early on. So the whole test took a couple of weeks, but the verification and validation, that takes a little more time.
And this looks like you're using isothermal technology rather than a thermocycler. Is that accurate?
No, it is a PCR thermal cycler. Our SARS-CoV-2 test is a real-time PCR test. The hardware itself will work with isothermal tests. So other groups that have isothermal applications could run them on our system.
What are the big advantages that this process has over other current testing options?
Yeah, I'd say the biggest advantage is distributed and fast. So as you can see, everything's really the size of the palm of your hand. It can be carried anywhere. It has an internal battery, so it can perform around 6 tests per battery charge. The consumables, as I showed you, are also very user-friendly, don't require any extra lab equipment. So you really can perform these molecular grade sort of lab standard tests very quickly.
And do you mind saying how quickly? So how long does it take for one sample, let's say, to get processed?
Yeah, for one sample, so the sample prep process takes a couple of minutes, loading the test another couple minutes, and then about 45 minutes for the thermal cycler to run. So, all in all, you're getting a sample done in about 1 hour. If you wanted to run multiple samples, it would add an additional 15 to 20 minutes on that.
And how many samples could you run concurrently if you wanted to using this process?
You could, yeah, you could run up to 9 samples concurrently on this thermal cycler.
And if you did do batches of 9, then it sounds like it would take maybe an hour and 20 or something all in.
Yeah, it would probably take about 30 minutes to do the 9 sample preps, and the loading them into the device and app is just a couple more minutes, and then the device running. So instead of an hour, it'd probably be about an hour and a half.
Got it. And you submitted this for FDA for review. When are you hearing back from them?
Yeah, so we submitted the test for FDA EUA authorization. It was first submitted by a clinical lab called One Health Labs, who used our products to develop an LDT, a laboratory developed test, and then submitted that to the FDA. Biomeme has gone ahead and also submitted, or will be submitting tomorrow, actually, a protocol, an end-to-end protocol. Really, the use case that we want to go after is more point of need there. So again, as I mentioned, that more distributed use case. And so the initial submission is from data collected in the lab, but we hope to amend the file and work with the FDA to demonstrate feasibility in a distributed manner.
If this does get government approval, which obviously everyone is kind of collectively hoping more and more tests are available on the market like this one, how quickly could you ramp up production of this?
So if we get FDA EUA approval, we've been working to ramp our manufacturing and scale that such that we can deliver as many tests as possible where they're needed. To do that, we've already initiated manufacturing of a second assembly line of our sample prep products. And we've been working with our manufacturers on the freeze drying side to also scale that. I think that work will be done in probably 1 to 2 months to really get to that sufficient scale where we hope to be able to supply thousands of these tests weekly.
Can you speak just briefly to the role of testing in general, how important it is to fight COVID-19?
Yeah, I think testing is critical to fight COVID-19. I think that the lack of testing early on really caused a lot of problems. People didn't know where the virus was, how much of it, what there was, and it really has led to an acceleration in the number of cases. You look at places like South Korea that implemented very good testing very early on. They've been able to flatten the curve better than we have so far. And so I think going forward, as we slowly thaw from the extreme social distancing and current lockdown measures, I think that testing will be critical to keeping the virus at bay, really understanding where it's at, who has it, and most importantly, track and trace individuals that may have been exposed, test them such that you can isolate. And I think that'll be how the world will slowly try to get back to some normalcy, from here.
Jesse, can you get a little bit into kind of where these devices and products would be best fit? Where are they going to be used in terms of like homes or clinics or hospitals in the US or abroad, any of those kinds of topics?
Yeah, so that really depends on the application or the use case for research or research use, as well as some of these other applications I mentioned, like industrial quality control. The tests are performed really anywhere. They can be on site, the back of a truck, a small sort of work office or a small lab. When it comes to the COVID-19 or the SARS-CoV-2 tests that we developed, the initial FDA EUA will be for use in moderate to complex CLIA facilities. So that's like, you could think of that as central labs, labs within hospitals, that sort of thing. We also hope to be generating some end-to-end test data of farther forward or distributed test settings. So small test, pop-up test sites. You could think of like, you've seen the drive-through collection sites. We see no reason that people couldn't be using our products right there on site to provide a fast turnaround.
One related question, Jesse, that just came up. from this? You said it could be done anywhere, including the back of a truck. That's a pretty powerful visual. Who can do the test? So, for example, do you need certain training? Can a patient do it themselves? That may seem obvious, but or do you need to be a clinician, or do you need to have some sort of lab credential to be able to pull this off?
I would say that really anybody with training can do that. I feel confident that I can demonstrate to a user more or less what I showed you and get them comfortable with the procedure. And then practice helps, right? So after a few times of them doing it, it really is pretty simple. In terms of the COVID-19 testing, I see the first users being people with a little more experience. So this could be lab technicians, medical technicians, nurses, those sort of users. We're not, it's really not in a place or approved for home use yet. So you won't see the initial products in the home. But one day that's where we want to go. And that'll be even more powerful when we get there.
Wow. To be honest, I went this whole interview and didn't realize that that was your overall vision. That's actually pretty incredible. Thanks for sharing that.
Yeah, no problem.
Can you speak to the sensitivity and specificity of the test and how it might differ from other tests that are out there at the moment for RT-PCR?
Yeah. So our test is quite sensitive. The LOD that we submitted to the EUA, that data package, had us at 1.8 RNA genome equivalents per microliter. So as you can see, that's right in line with many of the other EUA assays, molecular tests that are out there, the CDC, for example. In terms of specificity, we tested the test in the lab against influenza A, influenza B, RSV, obviously other common circulating respiratory organisms. We also did a full in silico analysis, which is what the FDA requires, where you look to publicly available databases to see if there's any predictions around cross-reactivity, and we were happy to report that we didn't detect any significant cross-reactions with a wide array of other organisms that we couldn't test in the lab.
That's awesome. Do you have any published sensitivity or specificity percentage, for example, an NP swab?
We also were able to get our products to a group in New York called Northwell Health. They're one of the largest hospital providers in New York. One of their lab directors and their team who do a lot of the lab testing has reached out looking for those distributed diagnostics. They did an initial evaluation with our end-to-end test against samples that had already been either determined positive or negative with other FDA EUA methods. And we found a 98% sensitivity. So we missed, there was one discordant result out of, I believe it was 42. So we're very pleased with that. It seems that it'll match most of the other EUA systems that are out there. I know that's a sort of small study for now, but it's with actual COVID-19 samples from NP swabs. So it was a very good demonstration of a capability.
What's your message for providers right now?
Oh man, keep doing what you're doing. You're critical. I think one of the best things that's come out of this is people really realizing and appreciating the healthcare workers out there, right? Who are working their butts off to make sure that as many of us stay healthy. And so I hope that that's one of the long-term lasting things that come out of this whole mess is a better appreciation for not only healthcare workers, but public health in general.
And then more broadly, what do you think can or should be done overall to raise the line? In other words, to expand the capacity of our healthcare system?
I mean, that's a tough one. I think that in our case, testing is critical. Not only companies like ourselves, but there's many, many others out there that are all working to provide as much testing solutions as we can. And it's going to take the combined approach of really everyone in the entire industry to do that. So I guess from my perspective, yeah, being more immersed on the testing side of things, that's where I see how we can help.
Jesse, it's been a pleasure chatting with you. I'm pretty impressed with what Biomeme's doing. I think our audience is going to be pretty wowed by what you just had to say. Thanks for doing it.
I'm Dr. Rishi Desai with Osmosis. Remember to do your part to flatten the curve and raise the line. Remember, we're all in this together.